Hello, my name is Johnathan, and I am a research programmer from Canada's Michael Smith Genome Sciences Centre at BC Cancer. Today I'll be presenting GoldRush, a de novo assembler for long reads with linear time complexity. Current state-of-the-art de novo long-read assemblers use the overlap layout consensus paradigm to assemble long reads. This paradigm consists of 3 stages: overlap, which involves aligning all the reads against each other; layout, which uses the alignments to generate a graph and traversing the graph to generate the assembled sequence; and finally, a consensus calling is computed on the assembled sequence. All of these assemblers have a high RAM cost. Here we present GoldRush, a memory-efficient algorithm with a linear time complexity. GoldRush consists of 4 stages: GoldRush-Path, GoldRush-Edit, Tigment-Long, and GoldRush-Link. We now define the golden path, which is an approximately 1x representation of the target genome. To generate the golden path, GoldRush-Path first initializes a data structure to store the reads in the golden path. This data structure is called the multi-index Bloom filter. GoldRush-Path then loops over the reads, querying each read against the multi-index Bloom filter. If the read is not found, the read is added to the golden path and inserted into the multi-index Bloom filter. If it is found, the read is skipped over and the loop continues. This iteration of reads is how GoldRush achieves a linear time complexity in the number of reads. To determine if a read is inserted into the multi-index Bloom filter, we use a read signature called the space seed. A space seed is a pattern of 1s and 0s, where 1s are k positions and 0s are don't care positions. In this example, the space seed design has 0s corresponding to the mismatched bases in k-mers 1 and 2. Using this space seed design, these 2 k-mers will be evaluated the same. The multi-index Bloom filter is used to associate the read signature with an ID corresponding to where the read signature came from. To insert a read, we first divide the read into blocks of length t times b, where t is the length of the tile and b is the number of tiles in a block. All read signatures inside a block are inserted with the same ID. To query a read, we first divide the read into tiles, and for each tile, we query all the read signatures and generate an ID and counts table. The ID with the highest count will be the representative ID of that tile. Once we have queried every tile of the read, we compile the results and give each tile a preliminary assignment based on whether or not the representative ID has an associated count greater than a threshold. In this case, the threshold is 10. If a tile is assigned true, then the tile is considered to be found in the golden path. Conversely, if a tile is assigned false, then the tile is not found in the golden path. After some more processing, we determine the query outcome of the read. In the first example here, every tile in the read is given a false assignment. This means that none of the tiles are found in the golden path, and we want to insert the read in its entirety. In the second example here, we see a mixture of true and false assignments. This means a portion of the read is already found in the golden path. In this case, we want to trim the read until there's a true tile overhang left and insert the trimmed read into the golden path. Finally, we have an example where all the tiles within the read are given a true assignment. In this case, the entire read is already inserted into the golden path, and we don't have to insert it anymore, so we skip over the read. After the golden path is generated, it is sent to GoldRush-Edit to polish and remove mismatches and indels. Once the golden path has been polished by GoldRush-Edit, it is sent to Tigment-Long to identify and cut any chimeric golden path reads at the chimeric breakpoints. Once the golden path has been polished and corrected, it is sent to GoldRush link for scaffolding. The scaffolded assembly will be the final output of GoldRush. To evaluate GoldRush, we compare it against 3 state-of-the-art de novo long-read assemblers using 3 different human individuals. On the left plot, we compared the peak memory usage against the time it takes to assemble the individuals. GoldRush assembles all 3 individuals within a day using a fraction of the RAM that its comparators use. On the right plot, we compared the NGA50 length against the number of misassemblies. GoldRush produces highly contiguous assemblies in the 20 megabase range with fewer or comparable number of misassemblies as our best comparator. In summary, we present to the community a novel linear time de novo long-read assembly algorithm that has the lowest RAM cost of its comparators and generates human assemblies with high contiguity and low misassemblies. I would like to thank my esteemed colleagues for their invaluable contributions to the success of this project. I would also like to thank our project funders. Thank you for attending my talk. If you're interested in trying out GoldRush, feel free to check it out at github.com/bcgsc/GoldRush or use the QR code. If you're interested in the stages of GoldRush, feel free to check out our posters at HiTSeq. I've also listed other talks and posters from the Bureau Lab if you're interested in our other ongoing projects. Thank you once again.